Welcome! Today, we are kicking off a two-part journey into the absolutely fascinating world of hearing, both the physics and the biology. We're going to start with a brilliant piece of engineering that's happening inside your head right now. One that solves a physics problem so fundamental that without it, you wouldn't be able to hear me at all.
So, what is this massive challenge your ear has to deal with every single second? Well, you can think of it as a fundamental mismatch, kind of like trying to send a message from one world into a completely different one. It is a classic case of physics just getting in the way of biology.
At the heart of this problem is something called acoustic impedance. Now, don't let the term scare you. You can just think of it as resistance, how much a material pushes back when a sound wave tries to vibrate it. Some things are easy to move with sound. Others, not so much.
And this slide really nails the huge contrast we're talking about. Air, where sound starts, has a super low acoustic impedance. Sound just zips right through it. But the fluid inside your inner ear, where the magic of hearing actually happens, that has an incredibly high impedance. It's like trying to get a sound wave to travel from an open field and pass straight through a concrete wall.
I mean, look at this number. Without some kind of solution to this mismatch, a staggering 99.9% of all sound energy would just bounce off your inner ear. It would be reflected away before it ever had a chance to be heard. And that translates to a massive 30 decibel loss in hearing.
So here's the bottom line. Without a fix for this problem, we'd be practically deaf to most of the sounds around us. Nature needed an incredibly clever adapter, a kind of biological impedance matching system to bridge this gap. And that amazing solution is the middle ear. So how in the world does the middle ear solve this huge 30 decibel problem? Well, it uses this really elegant three-part mechanical system. Each part adds its own little boost of amplification, and they all work together to overcome that incredible energy loss. It is an absolutely beautiful piece of engineering. All right, let's start with the first and, honestly, the most powerful of these mechanisms. It's a fantastic application of some very basic physics. It essentially turns your eardrum into a hydraulic press for sound. You probably remember from a physics class somewhere that pressure is just force divided by area. So if you take the same amount of force and focus it onto a much, much smaller area, the pressure just skyrockets. And that is exactly what the middle ear is designed to do. This chart just lays out the scale of that difference perfectly. The effective vibrating part of your eardrum, the tympanic membrane, is about 55 square millimeters. It collects all that sound energy and funnels it down to the tiny little stapes footplate, which acts like a piston on the inner ear fluid and has an area of just 3.2 square millimeters. This creates a massive 17-to-1 area ratio. Just by concentrating the force like this, the middle ear multiplies the sound pressure 17 times over. That alone gives us about 25 decibels of gain, which gets us most of the way to solving the problem.
Okay, so that was a huge piece of the puzzle. But we're not done yet. Let's talk about mechanism number 2. In between the eardrum and the staples are the 3 smallest bones in your entire body, the oscles. And they aren't just a simple bridge. They form a really sophisticated mechanical lever.
The first 2 of these little bones, the malleus and the incus, work together as a classic lever, just like a seesaw. The eardrum pushes on one end and the other end pushes on the stapes. It's simple, it's elegant, and it's incredibly effective. And the secret to this lever's power is the difference in the length of its arms. As you can see, the malleus arm is just a little bit longer, at about 8 millimeters, while the incus arm is a little shorter, around 6.5 millimeters. Now, this difference in length gives us a mechanical advantage, boosting the force by a factor of 1.3. It's a more modest gain, right? Only about 2 decibels. But hey, when you're trying to overcome a 30 decibel hole, every little bit counts. And finally, that brings us to our third and most subtle mechanism, which actually comes from the unique shape of the eardrum itself. This is where the design gets really, really clever. You see, your eardrum isn't flat like a drum you'd play. It's actually a shallow cone pulled inward at the very center. This curved shape allows it to buckle in a really specific way when sound hits it. And that buckling action concentrates the vibrational force way more effectively onto the malleus, basically doubling the force compared to what a flat membrane could do. And here's a quick clinical pearl for you. If any of these tiny mechanics get disrupted, it can cause some pretty significant hearing loss. Now, if you're finding this deep dive interesting, do us a favor and hit that like and subscribe button. All right, let's get to the final calculation. Okay,
Let's put all the pieces together. Let's see how these three amazing mechanisms combine to solve that problem we started with. How close do they actually get to our 30 decibel target? So, just to recap, we have our three amplifiers all working in concert. The hydraulic effect from the area ratio, the mechanical advantage from the ossicular lever, and that force-focusing trick from the eardrum's shape. So let's look at the numbers from our two main amplifiers. We multiply the amplification factors, so that's the area ratio's 17 times boost and the lever action's 1.3 times boost. That gives us a total pressure amplification of about 22 times. When you convert that to the logarithmic scale of decibels, it equals a gain of about 27 decibels. That, plus the extra gain from the eardrum shape, gets us almost perfectly to our target. And here it is the big takeaway. The problem was a potential 30 decibel loss. The solution provided by the middle ear is about a 27 decibel gain. It almost perfectly cancels out the loss, allowing sound energy to get across that otherwise impossible barrier. I mean, just stop and think about that for a second. This tiny, intricate system of membranes and bones achieves around 97% efficiency. It's a truly remarkable feat of evolutionary engineering, making sure the world of sound isn't lost before it even gets a chance to be heard. This elegant multi-stage solution is what allows us to perceive such an incredible range of sounds, from the faintest whisper to the loudest concert. It really is a mechanical masterpiece, perfected over millions of years of evolution. This biological machine is incredibly robust, but it's not invincible. So what happens when sound successfully makes it across this mechanical bridge, only to find the next part of the system is compromised? We'll explore what happens inside the cochlea in the next part of our explainer. We'll see you then.
All right, now continuing with mechanotransduction and the auditory pathway. We're going to explore the absolutely incredible transformation of a simple mechanical vibration into a complex neural signal. You know, something our brain actually understands as sound. Okay, let's dive right into this.
So we're going to start at the very heart of hearing. On a microscopic level, we're talking about the hair cell. This is the place where a physical movement so small it's almost impossible to imagine gets detected and turned into a biological signal. This whole process has a name, of course. It's called mechanotransduction. And you have to understand, this is one of the fastest, most incredibly sensitive biological mechanisms we know of. It's literally the conversion of a physical force into the language of our nervous system.
And when I say sensitive, I mean staggering. Get this. A deflection of just 0.3 nanometers is all it takes to open an ion channel and kickstart the whole hearing process. To put that into perspective for you, that is less than the diameter of a single atom. So the big question is, how on earth does this atomic scale switch actually work? Well, it's actually an incredibly elegant little mechanical system. So as sound waves make these bundles of hairs, the stereocilia, pivot back and forth, these tiny little filaments called tip links get stretched. Think of them like tiny ropes connecting the hairs. And as they get pulled tight, they literally tug open the ion channels, opening the floodgates for positively charged potassium ions to just rush into the cell. Okay, but for those ions to flood in so fast, you need a really powerful driving force, right? I mean, something has to push them. And the cochlea? It doesn't mess around with typical cellular gradients, no. It has its own built-in biological battery to power the whole thing.
So the secret here lies in two very different fluids inside the cochlea. Up top, where the stereocilia are, they're bathed in this stuff called endolymph. And this fluid is totally unique in the human body. It's packed with an incredibly high concentration of potassium. Meanwhile, the main body of the hair cell is sitting down in perilymph, which is, much more like your standard extracellular fluid.
So this huge difference in ions creates a really strong positive electrical charge in that endolength. We're talking about plus 80 millivolts. It's called the endocochlear potential. But believe it or not, that's only half of the story.
Because when you take that plus 80 millivolt potential outside the cell and you combine it with the hair cell's own negative charge on the inside, which is about minus 70 millivolts, you end up with this enormous total driving force of 150 millivolts. That is the power source. That's what makes this whole system so unbelievably sensitive and fast. Okay, so the cell gets flooded with potassium, it depolarizes, but now what? That signal has to be handed off like a baton in a relay race to the nervous system so it can start its journey to the brain. So let's take a look at how that crucial transfer actually happens. And this is a really critical moment of transformation. The hair cell's electrical signal, which isn't just a simple on or off, it actually varies in strength. It gets converted into a chemical signal. And it does this at the synapse by releasing the neurotransmitter glutamate.
Now this handoff, it doesn't happen at just any old synapse, oh no. It happens at a highly specialized structure called a ribbon synapse. And the best way to think about this is like a conveyor belt that's just loaded with vesicles of neurotransmitters ready to go. It is perfectly designed to give the auditory nerve a super fast, super precise, high-fidelity signal that can keep up with all the complex timing of the sounds we hear.
All right, so now the signal has been converted into action potentials, which is really the universal language of the brain. From here, it begins this rapid climb up through a whole series of processing stations in the brainstem, making its way to the cortex. Please like and subscribe for more custom medical videos made just for you. Now, moving on to the next segment.
So how are we supposed to remember this complex, but really elegant pathway? Well, there's a great mnemonic for it, E. coli. Yep, just like the bacteria. Each letter, or a couple of letters, is going to represent a key stop on this journey.
First up, we have E, and that stands for the 8th cranial nerve. This is the nerve that carries that raw auditory signal right from the hair cells and into the brainstem.
Next, CO stands for the cochlear nuclei. Think of this as the very first relay station in the brainstem. It's right where the signal hits the central nervous system and undergoes its first little bit of processing.
Then we have OLI, which stands for the superior olivary complex. Now this place is super important because it's the first stop that gets information from both ears. It compares the signals, looking at tiny differences in timing and loudness, and that is how our brain figures out where a sound is coming from.
The L is for the lateral lemniscus. This one isn't really a processing center. It's more like the information superhighway. It's just a big bundle of nerve fibers that carries the signal upward through the brainstem to the next stop. And the final letter in our mnemonic, I, stands for the inferior colliculus. You can think of this as the major auditory hub of the midbrain. Everything we've processed so far converges right here before it gets sent on to the higher brain centers.
So our E. coli mnemonic is great. It gets us all the way through the brainstem, but we're not quite done. There are two final, absolutely critical stops on this journey before we actually perceive sound.
Okay, from the inferior colliculus, the signal zips over to the medial geniculate body, the MGB, which is part of the thalamus. And the thalamus is basically the brain's main sensory gateway for everything. And from there, it finally arrives at its destination, the primary auditory cortex. This is where all those electrical signals are finally interpreted, and we have the conscious experience of hearing sound.
So when you think about it, this whole incredibly complex pathway, from a vibration smaller than an atom all the way to a conscious sensation, it all depends on every single link in that chain being perfectly intact. And that leads me to one last, kind of provocative thought. We've just traced this amazing journey, but what if just one tiny piece of it breaks? What if that super specialized ribbon synapse we talked about gets damaged? Well, there's a concept called cochlear synaptopathy, or sometimes hidden hearing loss. It means a person can go get a hearing test, pass it with flying colors, but still really struggle to understand what people are saying in a noisy room. It's a perfect example of how true hearing is so much more than what a standard test can measure.